So it's a pleasure to be here today, and I'm going to forewarn everybody that I'm going to talk about a bunch of different things. They all go together, and I think tell a very cohesive story, but we're going to talk about stem cells and neurons and glia and brain development and all kinds of things. So if anyone has questions or is confused, no hesitation to stop and ask questions in the middle. That's totally okay. I enjoy doing that. So I'm also going to start by doing my acknowledgments. I think this is something that is probably the most important part of this entire talk, and I don't want to rush through at the very end because I'm trying to squeeze in more data and not give time to this. So the work that I'm going to show you today comes from a bunch of different sources. My lab, a couple new folks who just started with me in the past year. The majority of the work that I'm going to show you is from work that was done at Stanford and Ben Baer's lab. Some new collaborative work from Dr. Serpushin here at Georgia Tech, and then a lot of work in the organoids from the Pashka lab at Stanford, and some other collaborators and funding sources. I also want to make a quick plug. So this is Ben Baers. This was my mentor when I was at Stanford, one of the fathers of the field of glial biology, and so very scientifically an important person in our field. But more importantly than that, Ben was a very important scientific advocate. And he passed away a couple of years ago from pancreatic cancer, but I encourage you all to go and buy his book. This is I'm getting no royalties from this. Ben wrote this book once he was diagnosed with pancreatic cancer. He knew he didn't have very much longer to live. He was a transgender scientist, so one of the very few people who understands what it's like to be in science as both a woman and a man, and how he was treated differently and how that affected perception of his own work. It's a very honest and wonderful story, and I think just discusses a lot about adversity in science. So I highly recommend this to anyone who is thinking about a career in academia or in the scientific field. Okay, so to jump into the work that I want to tell you today. So I am a neuroscientist, and as a neuroscientist, I'm interested in understanding this structure. And it's not just any brain, this is the human brain. I always give this introduction, and I always have to give some spiel about how deep down I'm an engineer, because most people that I'm talking to are not engineers. But now I get to talk to a bunch of engineers, which is very exciting. And I usually say, as an analogy, that I'm going to think of the brain as this puzzle, right? And I like this puzzle in particular, this Rubik's Cube, because there's many different pieces that you need to put together in a certain order to understand how this puzzle works. But in neuroscience, I would say for the past several decades, I feel like the way that we've been looking at this puzzle is we've been standing down here, staring at the face of this and saying, okay, we're going to solve this puzzle by understanding this one component of it, in this case, the red cubes, which are my analogy of neurons. But there's all these other pieces of this puzzle that we actually are not really investigating with the same depth and understanding. And so therefore, in reality, we're kind of confused about how these pieces interact with each other to form the complicated structure that is the brain. So, as engineers, really the way that to really decipher this would be to take this puzzle, to disassemble it into its pieces, ask ourselves what these pieces do and how they interact with each other, and in a perfect world, reverse engineer this model, take those pieces and build a more complex structure to understand how those pieces interact in development. Okay, so quick primer on what these pieces are and what I'm going to be talking to you guys about today that comprise the brain. So this is a general schematic of the major central nervous system cell types, and you may recognize some of them, and some of them maybe not so much. I want to go over them together. So highlighted in red here, we have neurons, which of course everybody knows about as the main signal transduction cells in the brain. But then we have some other cell types. So here we have the vasculature, which is comprised of endothelial cells, providing nutrients and waste removal from the brain. And then we have oligodendrocytes, highlighted in green here. So these are the cells that are myelinating axons in the brain. Then we have microglia, which are a very exciting cell type these days. These are the resident immune cells of the brain, responsible for dealing with infection and injury, and probably many other things that we don't know yet about. And then we have the cell type that I'm most excited about, which is the astrocyte. And here's a cartoon image of that reaching onto neurons and the blood vessels. So together, the word glia refers to oligodendrocytes, microglia, and astrocytes as a whole. There's no single cell type called glia. It's instead comprised of these other cell types, which vastly outnumber the amount of neurons in the brain. yet we understand very little about what they actually do. So this is an image of an astrocyte from a mouse. So this is a mouse where they've taken a pipette and they've patched onto this astrocyte and then labeled it with a dye that will actually permeate into the membrane of this astrocyte and label it green. In red here, we have these tubes. These are actually the blood vessels that have been labeled. So you can see that this astrocyte is wrapping around the blood vessels in these locations. That's actually as part of one of the functions that astrocytes do, which is blood-brain barrier formation. But this is just a very beautiful image, I think, and characteristic of what the cell looks like in the actual in vivo setting. So what do astrocytes do is kind of a fundamental question that we are still answering in the year 2019. For many years, we knew that there are these passive roles, and that's why the word Glia in German means glue. They were thought that these cells are just support cells in the brain. So they're buffering potassium and regulating glutamate and GABA transport, providing energy to neurons, and they do all that. That's all very important. But in the past about 10 to 20 years, a lot of really foundational work has kind of arisen that astrocytes have very active roles in brain development and function as well. So you need astrocytes to form synapses, to modulate those synapses, to even eat those synapses, to regulate blood flow, and there's a lot of speculation that these cells also serve as stem cells in the brain. So I'm just going to give you one example of one of these active roles. This is very straightforward. In this experiment, what you do is you take a bunch of neurons and you culture them in a dish by themselves. And so here you're seeing this neuron, and it's labeled in red. And then in yellow, what you're seeing are the synaptic puncta. So if you grow these neurons in a dish without any astrocytes around, you see very few synapses. As soon as you add astrocytes to this dish, or even if you just take the media that was secreted by astrocytes and dump it on those neurons in a dish, all of a sudden you get robust formation of synapses. And so in the past 10, 15 years, we've identified 5, 6, 7 of the molecules that are responsible for this process, but there's still a lot to be kind of discovered. How does it know when to secrete those molecules? Why are there so many of them? And how do they actually affect neurons to make more synapses? That's just one example. Okay, so where do you find astrocytes in the brain? So this is a pretty picture of neurons, but not astrocytes, in the cortex of a mouse using a technique called rainbow, which maybe some of you have heard about. Basically, all these neurons are just randomly labeled with a different fluorescent color. And other than making a nice image, it's just showing you that there's a lot of overlap and kind of all of these axons are crisscrossing each other and forming a very complex structure. If you take rainbow and apply it to astrocytes, called astrobow, you get an image like this. So this is the same area of the cortex, and what you're seeing are each astrocyte is randomly labeled either red, blue, or yellow in this case. So some of them are kind of squished up next to each other, but you can see that they're tiling the brain in this very beautiful kind of quilt. And so if you zoom in, what you can see here is the borders of 3 of these astrocytes and how their processes do not overlap more than a couple percent. We're still trying to understand why this is, but it's a very different architecture than what you see in with neuronal connectivity. And these astrocytes are actually connected to each other via gap junctions, just like cardiac cells in the heart. So the signals that are activating one astrocyte are going to pass through this syncytium to the others. Okay, I'm going to show you a movie now that after you see this, everyone will want to study astrocytes. It's not my movie. I'll put the citation up next. But a group recently from France was tinkering with new, interesting imaging modalities in the mouse brain, not too dissimilar from the rainbow that I just showed you. They're randomly labeling different cell populations with different colors, and then they're going to optically clear the brain and then image it with like some sort of light sheet microscopy. They ended up doing most of their work on astrocytes because when they were looking at all the different cell types in the brain, I think it was one of the most compelling and beautiful. So I'll just play this movie as they zoom through the mouse cortex. So here you can see all these different astrocytes labeled with different colors. And they're amazingly complex morphology. And this is only a subset of the actual astrocytes in the brain, probably about 5% of what you would actually see in the cortex. They're really incredible and very mysterious. So if anyone's interested, this was the paper that published this movie. There's about 15 supplemental movies that are all amazing. but mostly of glia and astrocytes in particular. Okay, so what am I going to talk about today? Now I've kind of set the stage for you a little bit. So how do we understand this? What we're going to do first is take this apart. I'm going to tell you about how we have efforts that we've undertaken to understand the molecular landscape of individual cell types within the nervous system. And the second part is going to be that reverse engineering, so using iPSCs to do 3D organoid models of brain development. Okay. And of course, the theme throughout all of this is going to be, I'm going to focus on astrocytes as a part of this picture because they are such an amazing cell type that we're interested in. So to start with this first one, why are we so interested in astrocytes in human development? I already told you that these are all of some of the roles that astrocytes play in development and function. So there's this easy game you can play, which is to say, okay, well, some of these roles are involved in processes that we know are related to disease. So synapse formation is involved in many different types of neurodevelopmental disorders. So is modulation of synaptic transmission. So is neuronal health and maturation. So is phagocytosis, and on and on and on. And so this starts to lead us to a hypothesis, which is, could actually primary astrocyte dysfunction drive neurodevelopmental disorders? Could it be that instead of being passengers to this pathology, it's dysfunction in these cell types that actually cause an aberrant neural circuit to form? So reason number 2 in support of that is that if you take, for example, a list of genes, so these are genes from the Safari Foundation, which is working with patients with autism. So these are the list of the most highly involved genes in autism within, I think this is about 2 years old. It's a whole list of these genes. If you take that list and say, what percentage of those genes that we think in some part drive the pathology of autism, what percentage of those are actually expressed in neurons or expressed in astrocytes? And you can do this with any disease you want, but I like autism. So not too surprisingly, about 76% of those genes are expressed in neurons, meaning that they could drive pathology in those cells. But what people don't realize is that almost as many, so 64% of those same genes are also expressed in astrocytes. And interestingly, 27% of those were only expressed in astrocytes, meaning we think this is a causal gene, some sort of impact in driving pathogenesis, and it's not even expressed in neurons, it's only expressed in glia. And we have no idea as of yet how that actually drives disease progression, but is why we are so interested in studying and modeling these cells. Lastly, I'm going to show you a picture of a mouse astrocyte here. So this is a section of the mouse brain, and this is a stain that's commonly used for astrocytes called GFAP, which is a protein that marks these major branches. So you're not seeing all the little fine processes of this astrocyte, but you see the major branches of this tree. If you use the same stain in a human piece of tissue, you see an astrocyte like this, same scale. So human astrocytes are about 10 times as large and about 10 times the volume of an astrocyte found in other rodent species. And it's not just their morphology that's unique. If you look at the molecular changes between rodent and human cells, if you take, for example, neurons between C. elegans or Drosophila, C. elegans, mice, and humans, the gene expression changes are very minimal between those species. But if you do the same thing with astrocytes or other glia, you see very remarkable differences over evolutionary time. And so we think that evolution is working quickly on these cells, and therefore we want to look in the human system and not just in mice. Okay, so how do we do this? In my lab, we use a technique that's been around for about 40 years called immunopanning, though I suspect almost no one in this room has probably ever heard of it. It's very simple. You take a petri dish, and on that petri dish, we just adhere primary antibodies. So these antibodies are specific to some cell type within the brain. So then we have your cell suspension that we've made from dissociating a piece of cortex from mouse or from human. And then when you pass those cells over the dish, those cells that are specific to that antibody will bind to the dish. You wash everything else away. It's like panning for gold. And this is a very straightforward method that we use. So this is an example of what that looks like when we do this in the brain. And we take a cell suspension, we have different antibodies for different cell types, and then eventually you have the cell type of interest, in this case astrocytes, that we want to purify. I will say this little green hepicam, this was 2 years of my life as a grad student, figuring out what that antibody would be that would work for human astrocytes. So this takes a lot of work to figure out what is going to be specific, but it's very effective. So let me show you some examples. So we started doing this with human fetal cortex. So this is a 17 gestational week fetal cortex. If we dissociate those cells into a single cell suspension, we can then pass them through these plates. So here's what happens if you take those cells and you dissociate them from the fetal brain, and you don't do any purification whatsoever. You just culture them for a few days, and then you stain for markers of neurons and astrocytes. You get a picture like this. So in red, you have neurons, and green, you have astrocytes, and they're mixed together, which is not surprising. You just basically took apart the brain and cultured it for 3 days. However, if you do the same thing with the cells that came off the neuronal plate, you get an image that looks like this. So there's one pesky astrocyte that snuck through here, but otherwise it's about 99% pure. And then you can do the same thing with the astrocytes. So this is a very effective way that we have of separating cells, and it has some advantages to other methods like FACS or flow sorting because it's so gentle that we can actually culture these cells afterwards. If you try to FACS sort these cells getting shot through a nozzle at 100 miles an hour, those cells will never survive to grow in culture. Okay, so we started doing this with fetal tissue, which is very exciting, but we were then interested in doing this in adult brain. This is a surgery that used to happen all the time. It doesn't happen very often anymore, but there are some patients who have a type of epilepsy caused by pathology deep in their hippocampus, which is right in this region. And at the time, the only way to fix this was to actually cut the hippocampus out of the brain. And if you did that, about 90% of these patients would be cured. But you can't just go in from the outside. The surgeons would have to come in from the front and remove this piece of normal, healthy tissue just in order to access the part of the brain that had pathology. And so they would give us those samples, and we brought them back to the lab as fast as we could. This is an example of what that looks like. So this tissue is not involved in the seizures in any way. We think it's as healthy as we could ever possibly get from humans. And we were able to do the same immunopanning to grow these cells from patients ranging from 20 years old to up to 80 years old, which was really exciting. Now here at Emory, we have another resource, which is the Yerkes Primate Center. And I didn't come here thinking that we would ever work on this type of tissue, but I was at a talk one day and someone told me that they unfortunately have to euthanize these animals somewhat regularly for various reasons. They have over 2,000 animals. And I asked them, like, what are you doing with the brains of these animals? And nothing. So we basically were like, oh, we can get these to the lab within 2 minutes. The lab, we just run them up the hill from where our lab is. And we now have a source of primate tissue that is as fresh as you could possibly get. But the advantage of the primate tissue here is that it's the entire brain. So we're not limited to one restricted region. So we've started some new projects looking at primate glia, specifically in macaques. This is just another example of this sample. It's really remarkable that this resource is here. Okay, so I'm not going to walk you through all of the things that we then tested about human astrocytes to see if they could do all the things that we thought that rodent astrocytes could do. But I'm just going to show you one as an example. So I already told you a little bit about synapse formation. So how do we actually measure that? How do we quantify that? So the experiment that we did is we took these human cells, we dissociated them, and then we grew the neurons on one plate, and then we grew the astrocytes on another, and then we put the astrocytes onto these inserts. And these inserts have tiny pores that are about 1 micron, so the cells can't actually fall through, but any molecules that they're secreting will be secreted down and fall upon the neurons, like raining down nutrients to them below. So then we can compare the neurons grown with or without astrocytes to see if there's a difference in the ability to form synapses. So here you can see we have 2 markers for synapses, a pre and a postsynaptic marker. You can kind of tell in the blue and the purple. So without astrocytes, the neurons beneath it, you're not seeing very many puncta here, so very few synapses. But as soon as you have these human astrocytes around, you see all this synaptic activity. So we quantified this through immunohistochemistry, and then we also performed electrophysiology to actually record from those neurons and say, not only are they making synapses at the level of these puncta, but they are electrically active and we can record from them. So then we went through and kind of validated all these other functions that we knew that astrocytes are involved with. So I'm going to take a quick side note to advertise for myself, which is that when we did this first experiment a few years ago, we were very interested in getting these astrocytes, but we also were collecting these other cell types from the human brain. And this was a very valuable resource for us. It's difficult to get human brain cell types. And we were performing deep sequencing, RNA sequencing on all of these. And so we had collected kind of a database of gene expression within the human brain. And so we wanted to distribute that to the neuroscience community, and we created this website, brainrnaseq.org, where basically, if you just go to this website and you're ever interested in where any gene is expressed in the human or mouse brain, you can very easily search for that gene and look at its expression. On the top in blue is mouse, and then the bottom is human. So you can see where these genes are expressed. And so this has been a very fruitful website, actually, in just disseminating data to the greater community. And I'd be happy to talk to folks later about this challenge of distributing otherwise complex data to an entire community and how to do that effectively to not overwhelm users to the point where they don't even want to use a website. I'm very proud of this slide. So this I took about like 2 years ago, but this is just the Google Analytics of this website that we created in 2014. So on top, you can see we created the website, and here you're seeing just sessions per day for a couple of years. But there's some interesting trends for the data scientists in the group. So firstly, the oscillatory nature of this is the fact that nobody uses this on the weekends. So we can, so it's a very weekday dependent. There's also some interesting blips here, January 2015, 2016, 2017. So everyone takes their holiday weekends or holiday 2 weeks very seriously and doesn't use the website. We're up to 105 different countries right now. I just think this has been a really fun experience in trying to distribute data to an entire community around the world. So it's been a fun thing to work on. Okay, so we found some interesting things when we were doing this work in fetal and adult human astrocytes. So not surprisingly, many of the genes expressed by both of those ages of astrocytes were similar. But there were some genes that were expressed only by fetal astrocytes or only by adult astrocytes. And we were curious about these. And some of them are very distinct. So here's a heat map of the top 100 fetal and top 100 adult astrocyte genes. They're very distinct. By having this signature, we can say you definitely came from an adult or you definitely came from fetal ages. So, of course, our question then is, what are these genes? Some of them are not too surprising. The fetal astrocytes express these proliferative genes because they're still dividing and proliferating. But some of these adult astrocytes were expressing all of these genes that we think are important for all those active roles that astrocytes do, for inducing synapse formation, for eating synapses, and controlling brain development in that fashion. So then we were curious, like, when does this happen? So the oldest fetal astrocyte we have is from 20 gestational weeks, the second trimester development. The youngest postnatal astrocyte we have is 8 years old. So we have a huge gap because we don't have much access to tissue during that those time periods. So the first thing we did is just a simple kind of data analysis of this. There were other groups that in the past had taken pieces of brain from fetal ages all the way up to 80 years old. Now this is whole brain, it's not just astrocytes, but they had a very high resolution of time points. So then what we did is we said, oh, we can just project our expression data of these fetal and adult markers on this expression data to get a guess at when this change happens. So here's an example. Here's a gene, aquaporin-4, that we knew was in this category here of adult astrocyte markers. So we look at its expression in the human brain across all these different time points, and we can see that it kind of turns on with time, which is what we expect. So then we do the same thing with another mature marker and another mature marker, and we can plot these. So here what you're seeing is all of the 100 fetal markers and all of the 100 mature markers plotted over time from that data set. And you can see that there's a very cohesive time point between the second trimester and about 6 months of postnatal development when all of those fetal genes are turning off and all of those adult genes are turning on. So we believe that this is a critical period of development, and especially glial development. But now we have a problem, which is that access to tissue during these developmental stages is almost unheard of. So it's going to be very difficult for us to actually start to interrogate what's going on mechanistically or biologically during those time points. And then if we were interested in studying disease mechanisms like in autism or schizophrenia, we're definitely not going to have the opportunity to study primary tissue at those time points. So those challenges are really what led us to the second part of what I want to tell you about, which is now modeling brain development in humans using stem cells and kind of switching gears. Any questions so far about astrocyte stuff? We're all good? Okay, awesome. So this is work that was done initially through Sergio Paschka's lab at Stanford, and then work that's being done in my own lab now here at Emory. So I think most of you are familiar with what an induced pluripotent stem cell is. Here's your 10-second refresher. You can take a somatic mature cell from a patient. You can reprogram it into a pluripotent stem cell, which by definition can become any of the major germ cell types of the body. And so in our lab, we're interested in brain development, and most of the cells in the brain derive from ectoderm. And so our interest is how do we take an iPS cell and push it to a neural ectoderm fate. So what was being done for many years, I'd say for the past like 10 years or so, until a few years ago, people would take these induced pluripotent stem cells, plate them down onto a dish, add some magic juju to them, wait a couple of weeks, and eventually you'd get some beautiful neurons in a dish. And this led to some really fundamental discoveries about differentiation and about human neuron function and in both health and disease. This was a big deal that we could do this. But there are some limitations to this method. So I am biased and care about glia, so we don't have astrocytes in this method. And because we don't have astrocytes, we don't have very much maturity of these neurons, and we also don't have synapses, because I've already told you those are some of the things that are dependent on astrocyte function. The reason that you don't make astrocytes in 2D culture is that the way the brain develops is that the first thing it does is it makes all of the neurons it's going to need basically for all of your lifetime. Once the stem cells are done making its neurons, then they switch to making glia, and that can take 5-6 months. It's impossible to keep these 2D cultures for long enough to actually see those glia come about. So instead, you just get these really young, immature cultures of neurons. The other problem with this culture, and I love this simple analogy, is this is what it looks like. This is amazing. These are iPSC-derived human neurons in a dish. And we're trying to use this to understand this organ. And so clearly there's a big gap in the architecture and the cell type diversity between cultures like this and then what we want to understand in the brain. So this kind of motivated us to think about, could we do this in 3D? So initially we had a method where we would take these iPSC colonies that grow they're like pancakes of cells on the plate. And all we did, this is not very innovative, honestly, we just peeled the pancake off the plate and we let it grow in 3D culture for the eternity of that structure. So it never has the chance to plate down in 2 dimensions. So we just keep it growing. We add a few molecules to powder into neural ectoderm. And then after many months or even years now, we have what we call an organoid, which can do a lot of different things that I'm going to talk to you about in the next couple of minutes. So here's some examples of what this looks like. So here's after 2 weeks of formation, you can see these structures are starting to form into these embryonic bodies. At 2 months, for any of those of you in the room who work with mice, this is one of our structures next to a E12.5 mouse brain, just for the sake of size. And then here's a plate of these. I always describe these to people as they want to know what it looks like. So if you've ever seen Israeli couscous, it's basically the same size and then consistency if you were to cook those. But basically imagine a lot of couscous in a dish. There's a new way, though, that we've been tinkering with to generate these. So instead of peeling off that entire colony, another option that we have is to take, this is an iPSC colony. I told you it looks like a pancake, so there's probably like 20,000 cells in this little pancake. And so then what we can do is actually dissociate that into single cells and put them into these things called aggrawells, which are just like little cavities, basically, where then over 24 hours, those cells will kind of aggregate into a very homogeneous sized 3-dimensional structure. And these aggrawells have been around for many years and have been used for all kinds of different methods and cancer biology and things like that. And then what we do is we just flush these things out of the aggrawell after 24 hours, and then we can culture them for 10 days or way, way, way longer. So here's a bunch of couscous in a dish. This is what they look like. When this first came out, there was a lot of interesting names that were given to these. And I bring this up because there's a separate discussion to be had about what this means to the lay public. but also because this field in general of 3D modeling is very new and has gone through a few changes in what we call things. So I'm going to call these forebrain organoids for the rest of the talk. When we first created these, we called them cortical spheroids, but the words spheroid and organoid and even mini brains and other things, they tend to refer to very similar biology if those are things that you've heard about in the past. Okay, so what's actually inside one of these things? What happens if you cut it open? So here's my 2-minute lesson of fetal development for those of you who don't have that on top of your head. So this is a schematic of the fetal human brain. At early time points, it doesn't have the gyri and sulci, so it's smooth or lissencephalic. If you take a section of the fetal human brain and cut it open, you would see something like this. You have a giant space here, which is the ventricle. We all have ventricles still in our brain, but they're much smaller than during development. And then you have a thin cortex that's developing on the outside. So then if we take and blow up a piece of this cortex, you can see down here you'd have the ventricle, and then you have basically the cortex going to the outer surface. And there are these cells down here that are positive for a marker called PAX6. It's not important that you remember that, but the earliest neural stem cells in the brain have this marker. So if we cut open an organoid and look, what we see are these regions that we call ventricular zones because they're PAX6 positive, there's neurons on the outside, and they almost have this luminal structure to them. And you can have multiple of these within a single organoid. Then you have my favorite cell type of the entire brain, even more than astrocytes, which is the radial glia. So these are the neural progenitor cells who live down here in the ventricular zone, and they have processes that extend to the ventricle and all the way to the peel surface, and they make this kind of scaffold. And they are stem cells. So when they give rise to neurons, those neurons will actually climb up the scaffold, and that's how they find their final resting place in the brain. So this is not an organoid. This is from a mouse brain. But if you actually look in the mouse brain and label these radial glia, you can see why they're called that. They have these beautiful radial processes, and you can actually see some of the neurons crawling up those processes in this image. So here's what it looks like inside of an organoid. You have, or the Death Star, whatever you think this looks like. This is one of these ventricular structures. Here you have these radial glia that are emanating from that process, and the neurons have basically used that as a scaffold to reach their final resting place. So this is all recapitulated within the structure. Because this is development in vitro, we can then also do some very cool tools and do things like live imaging. So the movie that I'm about to show you is we wanted to watch radial glia forming and doing things during development. So what we did is we used viruses that would either label neurons red or radial glia green, and then this is a 12-hour time lapse basically of one of these organoids that we just chopped in half and then put under the microscope in an environmental controlled chamber to watch the radial glia. So it'll loop a few times. So here you can see in green these radial glia reaching out to some undefined surface. Kind of let you guys watch this. I've seen this movie hundreds of times, and every time I see something new, there's all kinds of interesting cell division happening here. There's process pathfinding and communication between cell types. There's cells dying and moving. I think what's powerful about this movie to me is not that we've learned some specific thing from this time lapse, but the fact that we're actually watching human development in vitro. And this was a kind of remarkable and powerful ability within this structure. Okay, so just a little bit more to convince you that these things represent fetal development. So in normal brain development, you have these deep layer neurons and these superficial layer neurons that climb up the ladder, and they form a laminated structure. And although this can be somewhat variable within organoids, we do sometimes see really nice examples of that where you have those deep layers and upper layers separated in an architecture with the ventricular zone inside. And so we feel like this is recapitulating some aspects of the architecture of development. The neurons within these organoids are electrically active, so this is calcium imaging, spontaneous calcium imaging of a section of an organoid, and we can also record from these and actually just see what that activity looks like in these neurons. And here's an example of a method called array tomography to look at the synaptic structure and the abundances of synapses. So here in white are the neuronal cell bodies, and then green and red are all those synaptic markers. So there's synapses throughout this entire structure, and we can record from those as well. So I'm going to take 1 minute to say that a lot of those images I showed you were from a paper that we published in 2014 or 15. Since then, in the past about 5 years, there have been a number of different methods that have kind of been developed to make cortical organoids. And for the most part, they are pretty similar. Some of them differ in the fact that they are trying to pattern them to be different parts of the brain, like retina or midbrain or cerebellum. But the field has really exploded, and there are now a whole bunch of different protocols for doing this. The one thing that is starting to separate most protocols is whether something is what we call an undirected or a directed approach. So the very first organoid approach was what we call undirected. Basically, they form neural aggregates from these stem cells, and they just let them do their thing without any patterning whatsoever. The advantage is that you get a very complex structure. So these undirected organoids had forebrain and cerebellum and choric plexus and even an optic cup. So they had a very diverse set of brain regions. The downside of that is that it's really hard to do disease modeling when like one organoid has an eyeball growing out of it and the one next to it doesn't. There's just too much heterogeneity. But if there's other questions that you're asking that require heterogeneity, then that could be a very important protocol for you to follow. The protocol that I'm talking about here is something called directed approach. So we add a few more patterning molecules to very specifically choose one region of the brain to make. And the advantage of that is it's much more reproducible, the downside being we don't have the same diversity within an individual organoid. But what can you do with that? So here's an example of a beautiful study from Sergio's group. During brain development, there are two very different regions of the fetal cortex, this dorsal part in blue and the ventral part in red. All of the excitatory neurons that we make are going to be derived from this blue dorsal part. The inhibitory neurons of our brain come from the red part down here. And so what they did is they basically made organoids that were patterned to be each of these two regions. And then they said, what if we fuse these together and actually watch those inhibitory neurons from the red part migrate into the blue, which is the same thing that happens in real development. The interneurons that are born down here would migrate all the way up here into the excitatory cortex. And so here's an example of that, fusing these two different organoids together. They come together very easily after a couple of days. And then you can get an image like this. So in purple, you have your excitatory neurons. So this organoid on the right is patterned the way that I've been showing you. It just makes the purple ones. And then this organoid on the left is patterned to be ventralized. So it has both excitatory and inhibitory neurons. And those inhibitory neurons in green are migrating into the excitatory side. And so they use this to ask how that process was affected in disease. And it kind of starts to show you some of the power of this system when you start to put these pieces together with each other. Okay, so biggest question of the day, do we actually make astrocytes in these organoids? And the answer is yes. So here's an image of a 3D cleared organoid stained for astrocyte marker. Another image that I love. It also this one reminds me more of like the moon, maybe like craters in the moon. So you can see there's astrocytes throughout this entire structure. So the first thing that we did is we said, well, can we take this organoid apart just like we were before and watch astrocyte development over time? So here is just another image showing you that within a slice or within a single dissociated cell of an organoid, we've got these astrocytes here. And I can use that same immunopanning method that we used before to actually take an organoid apart and then separate it into neurons and astrocytes. This is the same kind of setup that I showed you with the primary tissue. So we did this, and the first thing we wanted to know was, do the neurons and astrocytes that we make in an organoid resemble actual human neurons and astrocytes at the genomic level? So we performed RNA sequencing, and these dots, each of these dots is a sample that we had collected from that first part of the talk when I showed you we were collecting human data. So not surprisingly, you've got human endothelial cell samples that look similar in oligos and microglia and astrocytes and so forth. So now we can project onto the same axis the RNA-seq data from our iPSC-derived cells, and not surprisingly, our organoid-derived astrocytes went over there and our neurons came over here. So this gives us some confidence that the gene expression profiles are similar amongst primary and iPSC-derived cells. Okay, but this was the main question, right? When does this transformation happen of a baby astrocyte to an adult one? We hypothesized that it was between the second trimester and 6 months postnatal time. So as a naive grad student, I'd say, well, it's fine. We'll just copy that in vitro culture. We'll just keep them for a very long time. And that's what we did. So here we generated organoids, and then we let them go for a very long time, almost 2 years of culture, which is, a very challenging thing to do, and especially would not be possible in two-dimensional culture. So then at each of those time points, purified astrocytes and asked what is happening at the gene expression level. I was excited about this, though, even before I get the gene expression data back, because when I dissociated these cells at these different time points, I already could see really fundamental changes. This is just under the phase microscope after taking these cells apart in the astrocytes. So here you have it. Early on, you have a cell that has no processes. By 590 days, just after dissociating these organoids, the cells have these tumbleweed-like processes still attached to them. So he's pretty confident that maturation was going on. So here's that same image I showed you before with those 100 fetal and 100 mature astrocyte genes. So then we could do the same exact type of assay by looking at those genes across developmental time within astrocytes. And you can see this transition happening exactly when we thought it would happen during the basically 300 days of development. So normally what I would do is take a few minutes to tell you about what that means for an astrocyte. So who cares that it goes from fetal to adult? And what we did is we did a bunch of functional experiments to see that when an astrocyte goes from being fetal to adult, it changes its ability to induce synapse formation, it changes its ability to eat synapses, and we think that it changes its ability to do a lot of other functions during development as well. I'm not going to walk you through that because I instead want to show you just a few minutes of new things that we're working on that I think might be exciting. But I'm happy to talk about those things afterwards if anyone has questions about the functional relevance of maturation. So what is the future of organoids? And when I say organoids, I'm specifically thinking about brain cortical organoids. So the first is this idea of using the system to study a term called molecular neuropsychiatry, which I'll talk about now. So this is a term that I think didn't exist a few years ago. The idea of being able to study neuropsychiatric disorders in a human model system was kind of an idealized thing that now we have the chance to do. So everything that I showed you so far used control organoids from healthy patients. But we can recapitulate any of these experiments using iPSCs derived from patients harboring whatever mutation or background that we're interested in. So we are starting with a few of these that are listed up here, but are also looking at rare genetic disorders and saying, how is brain development affected in these backgrounds? Okay, but the second thing that's most relevant to everyone here, I think, is this idea about engineering a more robust and complex system. This field is very new, and we're far away from where I think the potential for doing advanced brain modeling. And I want to talk about some of the areas that we need help in. So the first, well, here's a bunch of them. So I'll talk a little bit more in a second about these first two, about sophisticated patterning and about adding vasculature. Some of these other things are about introducing genomic engineering tools to the system so that we can manipulate it in vitro, doing multiplexed recordings at various different levels, somehow culturing organoids in a much more high throughput way so that we don't have hundreds of dishes of couscous in our incubator all the time, which is a very inefficient way and expensive way to actually grow these. To do assays in high throughput so that we can actually screen drugs or genetic backgrounds. Okay, I really like genomic engineering tools, apparently. It's on here twice. And metabolic assays, and I mean, I could go on and on about things that we need, but I'm going to tell you about the first two because we're trying to make some headway in this. So increased sophistication of patterning. So this is a new collaboration with a faculty member here at Georgia Tech, Vahid Sarpushan, his postdoc Martin, and a grad student in my lab, Sam. We were talking about this because in normal brain development, here's our fetal brain, the way that the brain is patterned along these different axes and what gives rise to forebrain versus the hindbrain versus midbrain, are the presence of these morphogen gradients. So these molecules called BMP or FGF, or Wnt and Hedgehog, and you may have heard of these before. And so during development, these molecules set up gradients of concentration, and the cells that then are kind of integrating that information to decide what kind of brain cell it should become. In our current organoid system, the way I showed you, we culture everything just bathed in a dish. And so there's no way to have sophisticated patterning. All we can do is say we want this entire organoid to be forebrain or the entire organoid to be hindbrain. So what Vahid's group is doing is 3D bioprinting with bioinks. And so our idea was, what if we could actually bioprint a 3D scaffold that we could embed an organoid in, and that scaffold could be doped or laced with whichever small molecule or protein that we want to create morphogen gradients on various axes. And so that within a single organoid, we could then pattern those axes in one structure. So this is an example of what that looks like with their 3D bioprinter. This is one of our first trials where you actually see the organoid here embedded in the middle of this structure. And so now the next task is basically saying, how can we add different concentrations of these concentrations of these molecules to create gradients within this cube, and therefore expose that organoid to different molecules during development. So that's intra-organoid patterning. There's also this idea that we could actually make like a snake. So imagine if you had a bunch of organoids lined up in a row. So you had high, in this case, retinoic acid on one side and low on the other, and you lined them all up. Could you actually then have a distribution of organoids going from hindbrain to forebrain within one structure that eventually could even fuse together? And so here's an example of that. Obviously, we are still working out the parameters of how to get these organoids in a nice single file line. But the advantage and the really innovative part of this technique is, in this case, we're actually 3D bioprinting the organoid itself. So these 300 micron newly formed organoids are mixed together with the bioink and then extruded from the bioprinter into this structure, as opposed to bioprinting something and then adding the organoid manually, like I showed you before. And then the other obvious extension of this is, what if we could add a vascular network to this? So organoids do not have an inherent vasculature, but could we use sacrificial bioinks like Pluronic here to print a chamber that could hold an organoid inside and have, and here you're seeing these blue cavities. So these blue cavities will eventually be filled with vasculature, but we're going to build a structure with this Pluronic and then basically remove it. So that looks something like this. You put the organoid inside, then you cover it up, then you get rid of the blue so that you're left with a cavity. You have your organoid embedded, and you have your cavities that could eventually be seeded with vascular cells. And here's an example of that. It's a little bit hard to see. You've got these cubes here. Each one has an organoid inside, and if you look really closely, you'll see these channels coming from each side of each face of the cube, basically. And so we're kind of trying to think about innovative and interesting ways to add complexity to this system. So that's why I put down here, and why I'm so excited to be here, is that being so close to a place like Georgia Tech means that there are a lot of really excited trainees and faculty who have interesting and new ideas about collaborating on this system. And organoids are so new. It really is about 5 or 6 years in its infancy that there's a lot of work that needs to get done to start to apply some of these techniques and make it a more robust system. So I'm just going to summarize for you, and then I'm wrapping up. What I hope I showed you today is kind of 2 parts. So the first was our work to take apart the brain and understand what those individual cell types did. I told you about immunopanning and how we can use that to separate cell types, and how we use this to learn about astrocyte maturation in humans. And then in our organoid model, that we've developed a system to model human development, that it makes astrocytes that recapitulate this maturation profile, and that now we can do interesting things to pattern it in much more sophisticated and careful ways. With that, I'd be happy to take any questions, and thank you for your attention.
Time for a few questions, please. Maybe you're like within your field, you're still working on that, but how do you maintain the concentration profile of the bioprinted matrix over a certain long time period?
Yeah, so that's the thing that we're actually actively testing right now. So that's going to depend on what molecule, if it's a small molecule versus an antibody. So like what's going to be the diffusion time and distance metrics that matter. But there are other options to make that fancier, right? You could use nanoparticles, you could use slow release molecules. We could change the architecture of the structure. So that's all in its infancy, and we're thinking about that question, but I don't have the answer yet. Yeah.
How does the organoid compare in size to the fetal brain at the same developmental stage? How does that affect the interpretation of the structures and the things that are going on?
Yeah, so it's much smaller. The fetal brain would probably outpace an organoid by about 3 months of development. And the reason then, of course, that the organoid does not continue to grow is that it doesn't have a vascular supply. So it doesn't have nutrients to actually continue to grow the way the fetal brain does. So I think because of that limitation, we never see the complexity of architecture of the cortex that you eventually see in the fetal brain. And it's because of that that most of our questions focus on those earlier time points. How are these different cell types established and set up during early development when it is more analogous? Because you're right. Otherwise, I think organoids, there's going to need to be some advances before we can actually try to model more architecturally sophisticated things.
Great talk. Thanks for that. So with respect to the disease models, you were showing specific diseases that had known genetic mutations. And I see a lot of people taking either patient-specific cells and having ideas to create disease models that way. So can you comment on, I'd like to hear your thoughts on doing the patient-specific stuff versus even inducing a disease in one of these organoid models that we don't know what the genetic defect is.
Yeah, so that's a great question and hotly debated topic in the iPSC field right now. And the word that gets thrown around a lot is isogenic. So those are kind of the two options. So one is just take patients, we don't know the mutation. The other is take the mutation and then correct it. So it's the exact same genetic background. That would be an isogenic line. And the field is flip-flopped on like which of these is the better approach. The scary thing about just taking patients, you say like, here, give me 10 patients who have autism and 10 who do not, is that there's so much genetic diversity that the odds that what you find is going to be related to whatever pathology is driving that disorder is pretty slim. When you think about what we do in mice and how inbred mice are, I mean, the genetic diversity in humans is so huge. I think where people have succeeded in that world of taking patients is when your controls are siblings. So this is sometimes done as well. So if you have brother and brother, brother and sister, or in a perfect world, twins, you can say, okay, we don't know what the mutation is, but we're going to take sibling A and then sibling B as the control, because at least then the genetic diversity is very minimal between them. I think the reality is that for most high-profile publications these days, you need to at least have an isogenic line, though, to prove that you've found some sort of single genetic cause. That might change, I think, in the future, but I think that it's hard to get published these days without being very specific, at least in one line, that you know what the mutation is. Yeah.
What about when it comes to like simulating maybe the periphery or signals coming from the periphery? So like the peripheral nervous system and its incorporation into the
Yeah, these are really great questions. So organoid cultures have been also generated from many other organs, right? So gut and lung and liver and things like that. I haven't seen much work on the periphery as far as like muscle, doing like NMJ innervation and things like that. There's a lot of interest though in doing gut-brain axis things. So if you take an intestinal organoid and you take a sort of cortical organoid and you fuse them together, could you recapitulate like microbiome level interactions between gut and brain, things like that. So I think those are questions that people want to do, but I don't think that the systems are existing yet for that. Yeah.
Have you done any 2D recordings from the organoids MEAs?
We have not done any from here. So you're saying 2D as in like take it apart, or did you say 2D?
No, I was kind of curious. Because it seems like you were planning on doing some ABA recordings.
Yeah, no, we would love to, right? It'd be a really nice way to do kind of readouts of what's going on if there is an activity level phenotype in any of these. We haven't done it yet. I think there's some technical challenges to making sure that would work, and you could think of some creative ways to even, like, there was a point we were thinking of, like, kind of embedding organoids on spires and things that could record LFPs from within, and so you can think of some cool stuff, but we haven't done it yet. Yeah.
Question.
Yeah.
I have plenty of science questions later, but
Yeah.
while the audience is here, you touched on like how this is perceived by the lay public, right? So,
Yeah.
like for a scientific audience, it's understandable to make an analogy to couscous or something, right? But there's other audiences where they don't see it that way, right?
Yeah.
They see it as like these are human-derived neurons, and you're growing a human brain in a dish. So I'm just curious about how you communicate this to a different audience, right? Because I know what I would say. And I want to know what you would say to say, like, look, we're not making miniature people in a petri dish.
Yeah. It's a really important question because I think that this is something that's been coming up a lot recently. I mean, even just a month ago, there was some talk about using this exact same protocol that we use and doing EEG recordings, and this idea that maybe there's consciousness in an organoid. So if I'm speaking to the lay public, I am always very, very careful to communicate that what we're doing is growing cells that resemble other cells that we see in the brain. And I use language like that. Like I would never say mini brain or like brain in a dish, because I think that is interpreted in a very misleading way to someone who doesn't understand like what is the debate about what consciousness is, and like, you know, where do we draw that line? So I tend to keep that in mind, and I do cringe a little bit when Wired or a magazine kind of takes this like a scientific article and then, you know, puts these labels onto it. Of course, we were never asking to use the terminology. So I guess the short answer is I am very careful to say we're just making cells that look like those in the brain. I don't say that we're making a brain or that we are trying to recapitulate development or kind of phrases that I did use here, because I think that the audience understands the differences between the two. Thank you, guys.